Yeah.
Calls, enthusiastic. That's motivated. Yeah. No, maybe that's true. Y'all study this weekend?
Oh, yeah.
Good stuff.
I think I forgot to tell y'all, one of the things I always tell people is the first 4 exams, study like your whole life depends on it. Like if you don't pass the first exam, that's it. There is no board. There is no, you're going home. Just think of it that way. Because the first 4 exams set you up so well for the semester. Did I say this before? This is an important thing. I really believe this. The first 4, I'm talking 1 for each class or so. Biochem, micro, A&P 1 and 2. If you get off to a good start on those, then your stress levels aren't as bad. Not that you still won't make it if you don't do as well, because you will, either way. But what I'm trying to say is they talk about all these study techniques, and your prior study may not work and all that. And just they say lean into that just for the first couple, as you know, my studying needs to be 10,000 times more than it was for any class I've ever taken for the first 4, and then let up. See what I mean? Because if you take them and you're like, wow, I just studied for 12 hours more than I needed to, well, fine, you're off to a good start. But if you're playing catch up, that's harder. Hopefully that makes sense. Yeah. Better than talking about mitosis and meiosis, but I'll just let you know, I was having a talk with my college sophomore's daughter, and I was talking about mitosis and meiosis. She just took over and started telling me everything about meiosis, and I finally stopped. That's more than my class needs to know. And I told her she's also studying forensics and genetics. So she's only a sophomore. I'm like, I know all that. She's, oh, we covered that in high school. I'm like, oh, boy. Finally got a kid that may be smarter than me. 5 of them, that's 1. No, I'm kidding, they're all pretty smart. They're all pretty smart. They don't follow my advice all the time, but they're pretty smart.
Maybe that's why they're smart.
Yeah, exactly. I like him. He understands good sarcastic humor. There's sarcastic humor, and then there's people who hide their ugliness by using sarcasm. Because what they said they really mean. That was good. I like that kind of banter. Keeps the day interesting. All right. So we left off talking about mitosis, and because we had a whole weekend where nobody probably looked really, really hard at my stuff. A lot of you were looking at A&P 2 probably more, but let's just, for the sake of the people who already got it, but the people who don't got it, let's start so we can refresh where we were at. Remember this, keep the cell cycle in your mind. Y'all need a recap, right? The cell cycle, little circle. So at interphase, mitosis is only forget reproduction. We haven't talked about reproduction yet. Mitosis just means that we know that cells get replaced, so a cell has 46 chromosomes, and it needs to replicate itself. And so when we talked about the cell cycle, you have a huge part of it that's interphase. So a cell is always, a cell that does mitosis is always, one of them is always in interphase, right? Remember, it kicks one out. So if it's in interphase, it goes G1, we start duplicating organelles, some of the cytosolic components. Then you go into that big S phase, and the only thing that happens in S phase is you replicate the DNA. You make an exact copy. That's a key word because we're about to talk about meiosis. But in mitosis, you make, well, you did, that statement's not going to fit later, but we'll leave it there. During interphase, you make an exact copy. Now, you go into G2, and that gives you the completion of cytosolic components, your organelles, and once all that's done, you'll hit the mitotic phase. And that's where, so interphase, it's not dividing. Then you're going to hit the mitotic phase, where you have prophase, right, metaphase, anaphase, telophase. Prophase is where the chromatin that was all relaxed starts to condense. So instead of looking like a big yarn ball or cotton candy, it's going to start to condense to ultimately become like we visually picture it, like an X or a line. After they condense, let's do this for the sake of argument. During this phase, right, we're going to say during prophase, will it help have the numbers with it? Right, during prophase, we have 46 chromosomes, but how many chromatids? 92. Remember, because we took one chromosome, and during S phase, we made a copy and attached it to it at the centromere, and we had two sister, that line's inverted. We have, you realize that. We have two sister chromatids, so there's 92 chromatids, but they're connected at the centromere, so we still have 46 chromosomes. Okay. Then we're going to go into metaphase. During metaphase, and I didn't go into this as much, but it is very important to note how they line up, is during metaphase, those sister chromatids or chromosomes, let's just say, are going to line up along the metaphase plate straight down it. So we only have 4 represented here, but there would be 46 chromosomes lined up straight down that dotted line. Tracking? Yeah, 46 of them straight down. Then you're going to go into anaphase. Anaphase is where we're going to pull them apart. Okay, so we're going to take the chromosomes that are lined up straight down the middle, and we're going to split the sister chromatids, and we'll pull. Right. I almost said that. We're going to pull them apart so that we wind up with 46 over here and 46 over here. Now here's where you got to stop using. Maybe all semester I'm going to be saying that. I need to stop using that script. Okay, so that everybody gets this. That's a key thing in people's minds that I know from teaching this. Is we're like, wait, you just put 46. I'm representing. What is this? Is this representing 46 chromosomes or 46 chromatids on each side? Thank you. Chromosomes. They were chromatids. I think I drove that home last week. They were chromatids when they were connected. So we had 46 chromosomes with 92 chromatids. Now we pulled them apart. Now we have 46 individual chromosomes in each cell. Y'all's faces and the way you reply makes you realize I didn't even really need to recap this, but somebody's like going, we talked about this last week? When did we talk about this? No. And then in telophase, right, there was one during anaphase. You remember when you go through cytokinesis, means you start to develop this cleavage furrow. After you pull them apart, they'll start closing in, creating a furrow, so ultimately we can separate the cells. That starts in late anaphase. That's in the previous slides. It's just not on this one. And then in telophase, you'll reform your nucleus, and the chromosomes will relax again and become chromatids. And at the end of telophase, cytokinesis will be completed and the cells will be separate. And so we wind up, we started with one cell that had 46 chromosomes, and now we have 2 identical cells that have 46. Whoops, sorry, I wrote 2. 46. Really messes somebody up. 46. It's hard to write with a mouse, and I was talking ahead of myself. So 46 and 46. That's mitosis. So once you get that, then we roll into how we pass on our genes and our inheritance. And so we do that through what's called reproductive division or reductive division. In other words, meiosis. Now, these sound the same. And because I use a lot of word differences in my mind, I'm from East Texas, Longview, so I have to say things in my mind differently. I often say them how they read. It's a redneck. Illinois. Anybody from Illinois? You hear me? I'm accentuating the S to show that I know that we don't pronounce that, but I always did when I was younger, right? Meiosis and mitosis sound the same, right? Especially if you're just learning it. Well, mitosis is duplicating a cell. Meiosis is making a reproduction. reproductive cells. And meiosis has an E in it. So I call it meiosis. So that I separate the difference, because under meiosis, we're going to have 2 rounds of division, and they both have prophase, metaphase, anaphase, telophase. There's a meiosis 1 and meiosis 2. So it helps me when people are asking me questions about during, they'll say during metaphase of meiosis 1, what's happening? Well, if I confuse that with mitosis metaphase, now we got a problem. Y'all aren't going to have that problem because of the way I'm going to discuss meiosis, but calling it meiosis sometimes helps. This is how it's spelled, meiosis. So here's what we need to do. Now that we know how to make a copy of a cell, what if we want to reproduce? So at the end of the day, right, when you take sperm and an ovum, they cannot bring 46 chromosomes each to the party. Not in humans. We could talk with bio and micro about different things, but not in humans. So we need the sperm to have 23 chromosomes, and we need the ovum to have 23 chromosomes. So when they come together, to reconstitute that, we get 46 chromosomes for human cells, right? And so we're going to form what's called gametes, however you'd like to pronounce it. But gametes are formed through the process of meiosis, right, which is a lot more complicated than just regular mitosis or mitotic division. So I just realized that I lied last week when I saw that. Y'all know what I'm talking about? No. Today. It will happen today. Because I lied. I said I would go fix that and repost it, and I did not. I need something to remind me. Never mind. I will. Somebody yell at me when I walk out of the room. Is that one cut off too? At the bottom? I can't tell. Let's see. And I know it says, but it won't be able to test it. Okay. Alright, so if we look at meiosis, this is what it starts to look like. You have 2 homologous chromosomes. We're in meiosis 1 now. So they are going to be segregated from each other, then you're going to wind up, my pen working again, with 2 haploid cells. Haploid means it has 23 chromosomes. Haploid cells are in. Gametes are in, meaning they have 23. Okay? There's a huge, I wrote similar on there, but meiosis 1 is similar to mitosis except for one huge difference. Right? And that is, we're going to form what's called tetrads. So we're going to take homologous chromosomes. Now remember, we have 23 from sperm, 23 from the ovum. Right? They come together. Each one of the sperm and the ovum each contains one of the homologous chromosomes. In other words, when you say what's chromosome 1 on the genetic map on any individual cell, you'd have 1 from the ovum, 1 from the sperm, 1 from mom, 1 for dad, kind of thing. And so they're sitting there. They're homologous chromosomes. We covered that last week. It means they look alike, they have the same linear sequence of genes, and they pair, remember this, during meiosis. So this is where they're pairing. When they pair up during meiosis, these homologous chromosomes form a tetrad. They cuddle. That's what I call it. They cuddle. And so those chromosomes that are normally sitting there during meiosis 1 in metaphase, they're going to get really close together and kind of wrap their arms around each other, just like they're laying in bed cuddling. In my mind. My crazy mind. Now, I want to make a note is I can say it on this. Crossing over is the reason why they form tetrads. Right? So during prophase 1, do this on your slide too. I'll tell you why I say this. Okay. prophase 1 of meiosis 1, they're the first stage, they're going to cuddle up. And during that tetrad formation, they're going to exchange or swap genetic information. So this is kind of different. There's a fancy definition there. The chromatids of homologous chromosomes, right, are going to form a chiasmata, wherein matching regions are going to break off and reconnect to the other chromosome. This is how we ensure genetic diversity. Right? So when I cut the slides way down just so I can talk about it, so you only have to worry about what's on the slides, then I can just explain it. So they're going to form up. The reason I told you to box prophase 1 is because when you go back to study it, what I know people do is they wind up picturing the tetrad. And they look like an X when they're in a tetrad, right? And so the book pictures, almost all of them I ever see, don't represent it well, because when you start thinking of it, and you're like, oh yeah, I get it. These sister chromatids came together and they swapped genetic information. See how they swapped some of them? Remember, they're the same linear sequence of genes, so they can swap some of that information from one chromosome to the next, or one chromatid. But you'll come back and go, oh, that happens in metaphase 1. No, it happens in prophase. It's just all the pictures depicted to look like it's happening in metaphase. Does that make sense? But I'm telling you right now, if I ask you when it occurs, the answer is prophase. Right? It happens in prophase 1. That's a key thing. Okay. Now, you can sit here and we used to do this, you can play with little pop beads and stuff, but I think people have a good enough imaginative mind to imagine that if two chromosomes come together and they form a tetrad, they can form a chiasmata, which allows them to exchange genetic information where you get a red and blue, and then on the far right you get one that's a mixed red and blue. This is why, if you want to know the importance, like everything that, if you look at your parents, right, you have all this info from one parent and all this info from the other parent. Do you look exactly like both your parents? A little bit. I mean, not really. Like my son looks 100% like my wife. Not a whole lot of, looking like me. Daughters are kind of different. It all varies because of this. Right? So if the better example is just using eye color. Your mom has blue eyes, dad has brown eyes. What color eye? We're going to talk about this. What color eyes are you going to have? Well, that's going to vary because of this. We're going to have diversity, genetic diversity. And that can be good or that can be bad. Because what color your eyes are really doesn't matter in the scheme of things. Whether or not you pass on a genetic disorder does. And that's included in this. Okay. So I simplified meiosis 1, believe me, down to, because I stated it, but I'm not going to ask you to tell me the differences between, I want you to learn all the mitotic phases, prophase, metaphase. I want you to know everything about those. In meiosis, I'm allowing you to simplify it down. There is, in meiosis 1, there is a prophase, metaphase. But all I care about is that you know that crossing over occurs in prophase 1. You don't have to separate them all out in meiosis. That's why I left them off. Okay. Then we go into meiosis 2. Meiosis 2 is going to now pull apart those chromatids. So let's back up for just a second here. In meiosis 1, we have prophase occur. Now, one of the key things that I need you to understand to get this is when I'm going to draw up here to represent that metaphase. So this will be representing this. Okay. How did, I'm asking rhetorical questions that I'll answer it. How did the chromosomes line up during mitosis in metaphase? Straight down through the centromeres, right down the metaphase line, or plate. In meiosis, they're not going to do that. Don't ask why. It happens. It's a fact, right? Forget about why. You want to study why? Go be a geneticist, right? Because if you look at the metaphase plate, when they line up during the metaphase of meiosis 1, they line up differently. They line up like this. So those same 46 chromosomes and 92 chromatids, right? We still have 46, 92. Why do we have 46, 92 in meiosis? What'd you say?
Starts with 23 in each gamete. So
But we're going to get to 23. You're on the right track. But what I was trying to get to something simple though is, what happened before you went into meiosis 1?
Still in the replication phase.
You still were in interphase. So interphase occurs before mitosis and before meiosis. Did I fail to say that earlier? So we started meiosis with 4692. Okay. So now we have 46 chromosomes, but they're going to line up like this. Yes, they line up like this. And I'm going to, just for the sake of argument here, I'm going to change colors for a second and say they're not really like that, because now they're like this. I'm just drawing to represent that they're different. And so they're a mixture because of crossing over. So if they're mixed like that, they're genetically different. So in this case, you put 23 chromosomes lined up over here, and 23 lined up over here. And so now when we separate them, at the end of meiosis 1, we're going to have 23 chromosomes in one cell and 23 in another, which is where the gentleman on the side was going to. Now, does that make sense? 46, we lined them up differently, we separate them, and now we have 23 chromosomes in each daughter cell, haploid cell. So you've completed meiosis 1. There's a second round, though, called meiosis 2. Meiosis 2 is where we're going to pull the chromatids into each of these haploid cells apart. So in meiosis 2, you're going to start with 2 cells that have 23 and 23 chromosomes. How many chromatids do they have? 46. Okay, so you have 2 haploid cells that go through meiosis 2. Now I'm going to draw metaphase again at the top to help you out. In meiosis 2 metaphase, they will line up this time just like they do in mitosis. They're going to do this. So that when we pull them apart, we're going to have 2 cells that have 23 chromosomes. She pulled the chromatids apart again. So I drew one of them up here, but remember there's 2 haploid cells that came out of meiosis 1. So down here we're going to wind up at the end of meiosis 2, you're going to have 4 cells that have 23 chromosomes each. All right, question. That's where the questions pop up. Key factors, if you're already understanding it. Key factor here is, once we crossed over in prophase 1, they're no longer homologous identical. That's why we can do this. And remember that during interphase, I kept driving home last week, during interphase, remember when I said there's a 2N and really we're 4N? To represent, we just have a double amount of the information. So we have 96 chromatids, 46 chromosomes, but it's just a doubling of the information. When you go into meiosis 1 prophase and you mix them all up, now they're all different. So it allows us to end up with 4 that have 23. It's making sense to everybody? Nobody has any questions on that?
So once gametes are broken up, like once they go through meiosis and they have 23 chromosomes, they won't like go through the process again. They don't always start out with the cell that has like 2 and break down.
Are you saying like once you form gametes, they will no longer go back into the cell cycle? Correct. Correct. They're going to leave as an ovum that's ovulated or as a sperm. Yes. Thank you. I should have said that too. Meiosis 1 prophase ensures that every single one of these 4 are not identical. You could also think of it as, okay, well, 2 of these say you're going to be a brown-eyed girl, and 2 of them say you're going to be a blue-eyed boy. Depending on which chromosome you want to look at for genetics, they're all jumbled up. So this is where we got to get talking about probabilities of what genetic traits. Yes, they're all different.
So if there's 46 chromosomes in the crossing over phase, is there only so many combinations then of different gametes? Or is it like an exponential possibility? And I may be getting ahead of myself.
It is. I wouldn't say exponential, but there's a wide variance because I say it's not exponential because whatever trait you're looking at, you only get one from mom or dad, or male or female biologically, right? Your mom and dad have blue or green eyes, right? So you generally are going to get one of those in there. The reason it's harder for me to answer that is because when you get further into genetics, your parents are carrying dominant and recessive traits that if you pass them, it can vary a lot. How about that?
Yeah.
It can vary a lot. In other words, can you have a trait, let's say, can you have a disorder that neither your mom or dad is suffering from outwardly? Yeah. You betcha. You can carry that. Females often, on that female chromosome or the X chromosome, can carry disorders and pass. We're going to talk about that. Anything else? I'll wrap it up for meiosis. I know it doesn't seem like it, but I tried to simplify it, so I'm going to recap meiosis. Because mitosis is you're going to get more value out of knowing mitosis than meiosis. But if you know this about meiosis, you'll be fine. In meiosis, we go through interphase, and then we go into meiosis 1. And during prophase, we line up our 46 chromosomes, and they all switch information. They form a chiasmata, whereas matching regions swap or cross over. Then you complete meiosis 1, you have 22 cells with 23 chromosomes. Those 2 cells will then go into meiosis 2, and at the end of meiosis 2, you wind up with 4 cells that have 23 chromosomes that are all individually genetically different. They're individual. What's your reference? That's what I want you to know. That's boiling it down for everything we just talked about meiosis. Yes?
So this may not be important, but please tell me this. So do they go immediately from meiosis 1 to meiosis 2, or is there an intermediary interphase where they replicating in the intracellular components, or is all of that taken care of during prophase 1? Or sorry, the
Interphase.
interphase prior to starting meiosis?
Yeah. Did y'all hear that? I'm terrible about repeating questions across the other side of the room and I don't hear it. But he's asking about does that process just continuous as you go through these stages, meiosis 1, prophase, and meiosis 1 and 2? And no, it's not. In males, it is. You're going to learn that in reproduction. Rosario is going to cover that. But you go through interphase, then you males enter meiosis 1, then they go directly into meiosis 2. So once males reach the age where they start producing sperm cells, from then for the rest of their life, they're going to continually go through meiosis 1 and 2. In females, when they're developing embryologically, they're gonna go through meiosis one, then it's gonna become arrested. After meiosis one, they're gonna stop. And then at puberty, they will start going through meiosis two and producing follicles that will become open. I'm trying to leave it there. So in females it's different. That's why it's a great question, but it leads you into repro stuff. But males it doesn't stop. Okay. Actually, all the possible follicles that you can ovulate for a female were there when they were born. Kind of interesting, right? But once they only, I'll leave it for repro. It's an interesting side of it. It helps explain why the later you have children, the more chance for genetic problems because things can start sticking together. I put this chart up there for anybody who really wants to hammer down the numbers. Like I said, as long as you understand the numbers where you're studying it later, that you're going to go, what is going to be meiosis? And you're trying to figure out how you got to 4 with 23. Here's what you're going to ask at home. I know I'm beating this to death, but it doesn't have to be that hard. I'm trying to simplify it. This is the take home that you're going to go at home and go, wait. You're going to go, meiosis, I started with 46 chromosomes. I made 2 cells of 46 chromosomes. That's easy because I made a copy of it during experiments. Right? Check the box, I got that. Then you're going to roll into meiosis 2 and go, okay, let me look at this. You're going to look at these numbers and go, okay, so this is just similar to mitosis. I started with 46 chromosomes, but I ended up with 4 cells with 23 chromosomes. And what I'm saying is, as long as you know that that is what happened, we're all right. Remember that when you're trying to, you're like, how did we get there? Well, he kind of showed you, but don't worry about how we got there. Because I just showed you how we got there, but it doesn't make sense to everybody. Right? All right. Yes.
When we talked about prophase 1 and the plasmata, I thought they weren't in tetrads yet, or it's just that in prophase 1 the tetrads hadn't aligned along the metaphase plate yet.
Correct. That's why I said box it off, is during prophase 1 is where the tetrads are formed. They just haven't aligned on the metaphase plate yet. So you could almost think of it, maybe that'll help people, I may be making this up, but this is probably true, is at the end of prophase 1 they already look like the ends. They just haven't lined up. I said it that way, and I like what you said. It is true. Alright. We got it. Now we can go on to fun stuff. We can talk about dominant genes, recessive genes. Hopefully we got all that. So inheritance. So when we look at a fertilized ovum, we're going to bring back the diploid number of chromosomes. 23 from one, 23 from the other, we get 46. And each one that came from the sperm and the ovum come back together, I said, and they're going to make homologous chromosomes that look alike, they have the same linear sequence of genes, and they pair during meiosis. And that's going to make what's called a karyotype. When you see chromosomes lined up, and they used to do this and cut them all out, and they line them up as close as they can along these centromeres, right? And so here you have one chromosome from sperm and one from the ovum. One from mom, one from dad there. You have 23 pairs, which gives us 46 chromosomes. And those are just all homologous pairs. When you look at the human karyotype, it's just a way to put them in an orderly arrangement, and they have 22 chromosomes and 23 are sex chromosomes. So then they line them up this way originally, and now they go back and look at each individual chromosome, and they've constantly been mapping what's on each chromosome. In other words, what genes. Now here's the real thing, is when you take two homologs or homologous chromosomes, and you look at them, I want you to remember, I keep repeating it, they look alike, they pair during meiosis. I keep saying them different ways, I'm saying the same thing. And the last part said that they have the same linear sequence of genes, right, that control traits, or how we make certain proteins that do things. But they're going to do so with variation. So the best example of this, once again, is eye color. They're called alleles. Okay. So on each chromosome, one from sperm, one from ovum. I like to use mom and dad. I know we don't like to hear it that way sometimes, but you say mom and dad. And mom has a chromosome, well, let's say we're looking at chromosome 1, and it's where we find eye color. Eye color, the genes that code for eye color are on the same location on each chromosome. One from mom, one from dad. Brown, blue. They come together. But where the location is for that genetic portion on each chromosome is the same, but depending on the instructions there, one's going to code an allele for brown eyes, and one's going to code an allele for blue eyes. So alleles are the same genetic makeup, but they're different expressions of it. That's what you would call it. So you would say, well, they have different alleles. They're not always different. Depends on what the parents do. And so if you had somebody who had homologous chromosomes, right, that have the same alleles, you would be big A, little a, or big A, or big A, little a. That's homozygous. So I'm going to keep using eye color. Both your parents have dominant colors for brown eyes. Right? When you look at that offspring, they're going to have big A, meaning they're going to have brown eyes because their parents are homozygous for that gene. And that could be on, you can have two dominants or two recessive. Doesn't really matter. It's going to be expressed. We're going to look at probabilities. What's going to be expressed? If an individual has different alleles, they're going to have big A, they can be different letters. We just use these to represent. Big A, little a. So they have different alleles. Right? One from mom, one from dad, but they have different alleles. But the dominant, if there is a dominant allele, it's going to be expressed. So whatever the dominant one is, will be expressed. Okay, so that's heterozygous, meaning they're different. Another concept you look at is when I look at any individual in this room, if we were to pull up your genetic map, we would see that you have a genotype, and we could map out all of your genetics. Everything that's in your genetic code, that's your genotype. Your phenotype is what you're expressing. So go back to eye color. Anybody in here's parents have different color eyes? Yeah. So if your parents have different color eyes, let's say you have brown eyes, right? You have brown eyes, which parents both don't have brown eyes. You're heterozygous for that trait, for eye color. You're expressing whichever one's dominant. Right? So the dominant one's being expressed. Whatever you're expressing, so if your mom has blue eyes and your dad has brown eyes, and you have brown eyes is the dominant, and that is your phenotype. Your phenotype includes brown eyes. But your genetic includes what? Your genotype includes brown and blue. You're heterozygous. Okay? One's dominant, one's recessive. And that's what this slide talks about. Anytime there's a dominant allele, right, it's going to mask the recessive allele. So, in other words, if you have something in your genetic, your genotype, that you're not expressing, then there's a dominant allele causing that to be repressed. You're not seeing it. So you can have a person who's a heterozygote that has a different genotype from a person with homozygote. You can have somebody who is big P, little p, just leave it as eye color. Big P's brown eyes. And you can have a person who's a homozygote who has big P, but they both have the same phenotype. Why is that? Because big P is dominant. And it doesn't matter if you have two big P's, you're not going to have browner eyes. Well, you could, I mean, but you know. Right? You don't get a double dose of brown. Well, maybe you do. I don't know. I'm not a geneticist. But that makes sense? At all? Dominance will always mask the recessive. In the beginning, that's where we start. So why do we study this stuff? It's interesting, like you start thinking about your genetics, okay? But really, we have to do that to understand things like the ABO blood group system. There are a lot of blood group systems, and we're just choosing to talk of one as an example. And because everybody in here in the military generally knows their blood type. And you ask my kids, they may or may not know their blood type. I'm not sure if they do. I hope they do. But military, we're always pretty aware of it. And that's where we get A, B, and O. Everybody in this room has one of those blood types. And what that means is antigens and antibodies. So if you look at somebody, a blood cell who has type A blood, we know that there are A antigens on their blood cells. This is going to drive me nuts by the end of it. We'll fix it today. Okay, I'm not going out of my office until I fix it. Okay. I had to pull that up because the bottom's important here is A and B are codominant when you look at the ABO blood group system. Both A and B are dominant to O. So if you have an A or a B, it's going to be dominant. If you have an A and a B, they're going to be codominant. If you have an A and an O, you're going to express the A because A and B are both dominant to O. So let's look at what this means. It'll make more sense. Here's the ABO blood group system, and if we say we have type A blood, alright, so I'm A positive, so I have type A blood. That means that when I was born, I already, because of genetic makeup, I already have A antigens on my red blood cells. And after I'm born, due to environmental factors, because I have type A, I will start to develop anti-B antibodies. I will develop antibodies against the B antigen because of the environment. Because I don't have that. So I'll develop antibodies against it. If you have type B blood, you have the B antigen on your blood cells, and environmentally you'll develop anti-A antibodies. If your blood type is AB, you've got an A and a B from mom and dad, so you're AB, you have both. Remember, they're both codominant, so you have to be AB, meaning that you'll be presenting both the A and the B antigen on your red blood cells, and because of that fact, you won't form any antibodies, because you already have both. Remember, we're just looking at one blood group system. Don't think past that. And then you have type O. Type O means you were born with neither A or B, and you have no antigens, so you will develop antibodies to A and B. So these A and B antigens are what we see in your glycocalyx, to bring that back. Cell identity. So these antigens identify your red blood cells. So the antibodies that are created, I put it down at the bottom of that, the antibodies that are created, like I was born and I had A antigens on my red blood cells. And I'm gonna develop anti-B antibodies due to the environment. And that's going to protect me in the case, if I was to get type B blood, is that gonna be a good thing? No, I'm gonna have a transfusion reaction because my body's gonna go after and kill those B antigen blood cells that you put in me. I'm gonna have a big transfusion reaction. Can you, I'll finish out with, as we transition to this, can you give somebody blood types that aren't their normal? Can. And I have, with controlled environments, given people, with the doctor I was working with, blood that we had to give them different blood type, but we also had to just watch them really closely. It's like saying, can you give somebody penicillin who's allergic to penicillin? Yeah. Should you? No. No, not yet. If somebody's allergic to penicillin, and let's say, and I worked in spinal cord injury, I'll use this to take us into the break. I worked in spinal cord injury, and we have urinalysis. Every day my job involved working with wounds and reading urinalysis reports and figuring out what is this strange sounding bug they have in their urine do. And we'd have terrible looking urines. They're coming out of leg bags and things. And so they often had UTIs, urinary tract infections. But they have like multi-resistant antibiotic type UTIs, right? If you've ever seen a lab report, it's just huge for the urinalysis on it. And I'm having to figure out with the pharmacist what antibiotic we can give them. And we ran into a couple of cases where we couldn't clear up a UTI and they're either going to bank or something. Anyway, they wanted to give this person, I didn't come up with it, I'm not that smart. They said, we're going to give them penicillin. And I'm like, at this high dose, whatever. I said, well, they're allergic to penicillin. And this is the first time we did it. And he said, yeah, we're all going to be there. And we had everything set up, ready for if they went into anaphylactic shock. But we also, each dose we have to go over there for the first few, but we titrated it up very slowly and wound up getting rid of that bug. Some of the bugs they just live with forever. I need to bring in if you've ever done urinalysis and seen like, oh, you got a bug in your urine, pull up one from spinal cord and you're like, holy crap, what's in your urine? That you get used to. All right, let's take it.
So there's two. You have like blood type and then you have RH. So that's the different part.
There's more than, we're going to talk about RH for a second, but RH is positive.
Alright, sir.
Oh, you got that slide up, so I know where we're going.
So, that's what I was trying to get at. So that's just way too deep, right?
Yeah, just assume they've already pulled it apart. You had, I knew at the end of that class, I probably knew what you were saying.
Okay, and then you keep saying about like heterozygote. So if you have heterozygote with the homozygote, you're always going to get dominant. And this one has recessive cells. If you have two heterozygotes, you have recessive cells. It's like if you have two parents that are from, they're heterozygotes.
You have two parents that are heterozygous.
It is possible that it's super close.
25% of the time if you look at like
All of that is made.
And actually, when we look at things like hand, number of fingers on our hand, we are all, we have both recessive genes. dominant trait in our whole gene pool, we would all have 6 fingers.
Is that just something that has been through evolution?
Somewhere in I've had one in here. In the class, 2 classes ago. There's a famous picture you guys see. That's just a little bit unfair. You
Yeah,
get a little
because you
extra
can grab a
grip on
lot
it.
and get that curveball. Well, you have the grip on it.
Well, that's what I was thinking.
Six-seat fastball. Yeah. You just grip the ball like this and throw your knuckleball. Instead of gripping with your knuckles, you throw it like this.
It's only one. There's only 46 chromosomes. Yeah, 20. Which one of those? Yeah, 46.
This one I'm excited to do. This one I'm excited to listen. How big the correctly have
I thought you were saying like you're gonna go in miles and one. I thought you said I'm gonna take one time so don't worry about it. And how does that make a difference? It makes a big difference. Like I was thinking that I can pass it on.
Yeah, I was watching, I was like, next level. Other side was technically to the right, like right, people on this side is right, versus like, you know, it's like tails on the side, which is, yeah. That one's usually less as well. I mean, also just obviously, you know, easier. I just sit, we're all waiting for like 2 minutes to work, like an hour. Well, the other one is kind of yelling at everybody in the bathroom. And then back to the and then I run out. Nothing wrong with that at all. thought you were gonna start pressing. Jimmy. Hey, I got a weird guy over here. That's crazy that you didn't invite anybody from taking up capital this weekend. I'd love for you guys to come and eat my free food and I would buy it all.
Yeah, they didn't. I just showed up with those cats.
He actually had somebody choose the he didn't invite me. He did more than just so poorly in the house.
Well, really, my house is left.
Probably not. I'm not going to send you guys out. So I did too. But you want to face the rejection. But now that I know that we're like saying all the time, we know everyone. Now, retroactive, yeah, crazy. I always felt like I was it would have been the right motivation. You don't have the right motivation. I got it. You didn't invite anybody? Yeah.
We had our little group. We showed up for they showed up like right as the day started or afterwards. And we did it in the morning, but how did we have our little morning huddle? And then they're like, and then all of a sudden, like, the J3 they started getting mad at me. Like, they were going to take away my rent and make me do it. It's like, first of all, you guys were like good at work. It's like,
Wow, did you use that?
of course not. I just said sorry. You can be there for my next one.
I was in my unit for like 4 minutes. I showed up, like great, lots of people, and like I came from the route or whatever, but I had to put it on. Coloring a bar in.
Literally, Sharpie in the dead in front of my boss. He was like 6. As a captain, I told my boss,
Dude, you got away with I had 0 because I had sinners too. There were people who like loved it. You know, like you were kids who were like, oh man, I don't have enough time. That's cool. We do. Oh yeah. They made a couple of washes, Matt Sharpie, quick fade. Color it back.
I didn't know it. They were showing me this.
That's awesome. That's awesome. That's so awesome that they also took them off the game. We got first consideration. We got campaign. That's awesome.
PFCs.
PFC colored in.
Colored in like special. It wasn't
That's like that's like real uniform. You just describe your days.
How far did you make it in the list from sergeant?
Whatever happens, you go right to sergeant. Maybe. I always feel so bad. It's like they don't really pay attention.
the hardest thing to do is when you meet a corporal
And they're like, yeah, it's hard way.
I saw one guy with cold rings on his back, on his recording, so I was like seriously super hard. Okay.
Who were you with at Bliss?
I was in
Second brigade. My brother's in first brigade, and they're next door neighbors. He was in 163. I started out in Charlie Bear.
All the same path.
My one brother, he's a infantry. My brother enlisted infantry. My brother went to civil affairs. My brother's a bachelor of it now.
Oh wow.
I left school. I left school. And my brother just
No way.
What's your branch now?
Last year they did like you were like, gotcha. Ah. Yeah. No way. Stop. He said.
We got in trouble, it was like
I didn't
push-ups.
get my fill in this weekend. Parents. When you were younger, how long did it require?
All right, here we go. I should have finished this slide before we ended.
You know what I mean? Like, I feel like that's kind of like that progression.
If you want to be a lab person, which probably none of us do anymore, we want to be PAs, but this is a good example. You want to figure out blood group types or blood types, right? And I talked to a few people with a break about some blood stuff. Right, we're scratching the surface, as they say, on blood group systems. We're not getting that deep. But if you took a drop of blood and you did not know what was in it, or sorry, what blood type it was, or what antigens were there, you could take a drop of anti-A, or antibodies against type A antigen, drop it in that blood sample, and it would agglutinate or clump up. That would tell you that person's type A. You took antibody A, right? It took antibodies against the A antigen and put it in that blood sample, and it reacted. And B would have no reaction. And so you could follow this chart down all the way and say, well, if you gave them B, it would clump up here. Now you're going to drop both drops in there, right? One at a time. Different drops, so you know. Because if you don't, you wouldn't know that they could be A and B, AB, because they're codominant. Or if they have no reaction to either one of the serum, you're blood type O. That's called forward typing. There's also reverse typing, but we're not lab techs anymore. So I say anymore for the people, the few that may be in here. Another illustration of alleles, genotype, phenotype. Remember, keep in your mind, genotype is all your genetics. Phenotype is what you're expressing. Right? Genotype is everything that came from the sperm and ovum, mom and dad, however you like to say it together. But then what you have. You may have a genotype for big nose and little nose, but you're expressing little nose. You still have genetic material for big nose in you. Okay, but let's look at dwarfism. So dwarfism is a great example of looking at dominant and recessive alleles. So when we look at dwarfism, we're just using D to represent it. The alleles for dwarfism, the dominant is big D. Normal height is little d. So what do we say the dominant does? Dominant always is expressed phenotype. So if in our genetic code D is big D is dominant, how come we don't all have dwarfism? I know some of y'all know genetics and you're like, come on, this is too simplistic, great statement, but this is helps you understand. Well, it's the difference between genotypes and phenotypes and what we see in our genetic code. Okay? They study it. And so when they study it, you look at, because if somebody has big D, that means they are homozygous. They have both the dominant alleles for dwarfism. That's very severe, and truthfully, they're probably not going to live more than a year. But if you have a big D, little d, in that case, the big D is going to be expressed, and you're going to have dwarfism. But all of us in here are little d, little d. So in this case, we're homozygous, right? Recessive. We have both recessive. So in our gene pool as a whole, we don't normally have big D. So that begs the question now, if two of us in this room, I say this room just to kind of bring it home, or two adult, male and female, in this room have a kid together, will they have a dwarf? Or can they have, that's negative to say it that way, can they have somebody, a child, an offspring with dwarfism? Not genetically doesn't seem possible, but that's how it happens. In other words, can normal height people, you ever watch that TV show? Did they have someone had normal height parents? You didn't see the parents on it. Normal height people can have a child that has dwarfism. But a normal height person has two recessive genes. So if they both have little d, and little d, and they meet up on Friday night at Cowboys in San Antonio, is it possible for them to have a child with dwarfism? Yes. Yes. Why? That's why I said this is a great example. Why? Okay. So the most common cause of dwarfism is achondroplasia, which is a type of bone growth disorder. And 85% of the children that have achondroplasia are born to average-sized parents. Little d, little d. And so since achondroplasia actually falls as that big D, one of the dominant genes for dwarfism, it happens by what they call a de novo mutation in either the egg or the sperm. Okay, de novo is a fancy word that in East Texas we pronounce they don't know. Right. Anytime you see de novo, it's medical scientists saying they don't know. So I just say they don't know. De novo. They don't know. So you have a mutation that occurs, and next thing you know, you got a dominant allele for it. Now, we could finish it out just for fun and say, and two individuals with dwarfism have a child that's normal.
Yes.
Yeah, because they're heterozygous. The probabilities on a Punnett square would show you that. They could pass on two recessive genes and have a kid that's normal height. So interesting stuff. I hope you can see why that's a good example. Another I corrected myself very quickly. Not funny about it. That was a play on words that I didn't mean to say. Another interesting fact is when you look at how many fingers we have 5 fingers on each hand, right? 5 digits. And so the alleles that code for normal traits are not always dominant. Right? So the gene that codes for 6 hands on the fingers is dominant. We don't have 6 fingers. I've had a student that had 6. I've had 2 students actually that had 6 fingers. Oftentimes it's not a full other finger. It's like a nub. And so most of us are homozygous recessive for hand dominant, how many fingers we have also. And so we mention all these so we can look at the probabilities. You've heard of all this genetic testing now where they say, well, you can actually go and test mom and dad, mouth, whoever you want to look at, and you can show up at the bar with your list and go, yeah, I'm carrying these genetics, and you can match them all up. Find somebody you want to pass on with. You can do genetic testing on a lot of things, but I don't think that's a great idea. You do it if you want. Okay, so when we look at how we're going to figure this out, we're going to look at a Punnett square. And then a Punnett square, we're going to line, whoops, I didn't put it like that. We're going to line them up. You're going to make a box. So you're making this box with 4 squares. And over here, if you take, it says eggs, but ovum. Okay, so you have big A, little a. So both parents are heterozygous. Big A, little a. And the sperm over here, you have, right, big A, little a. So we're going to use the Punnett square to map out the genetic possibilities of the offspring. So if we were to do this, I think I have fly-ins here on left. So we'll just, if you have big A, that means that you're dominant for whatever trait that is. You're homozygous dominant for that trait. You will express whatever A is. If you have big A, little a, heterozygous. If you have big A, little a, once again, you're heterozygous. And then you have little a, little a. Obviously, everybody in here is intelligent. They understand that we're just dropping these down to match them up, right? So that we can look at the probabilities. So what we want to know is what are the possible genotype of this individual and what is the possible phenotypes. Those are 2 different things. Okay. So if you look at this Punnett square in this example, the possible genotypes are, you can have a genotype that's 1 big A, you can have 2, right, that are heterozygous, big A, little a, or you can have 1 that's recessive here. And so then you go, well, what's the probability that we're going to express certain genes? Okay, so let's use I-color. A, we're going to say that A, whoops, just trying to drop, equals brown. Brown eyes. And little a equals, let's just say green so I don't have to write B. I was going to say blue again. So equals green. Now green eyes are a whole funky different stuff. You can talk about somebody else, but green. Because blue eyes is interesting too. You know they've mapped it down. Do they say some person in Russia 10,000 years ago had a genetic mutation that caused people to have blue eyes? But it's not actually part of our regular gene. Who said it before the break? Somebody came up to me and asked me, talking about cheers. That's Cliff Clavin knowledge. I know you don't know what that means, but some of y'all do. Okay, so now if A is dominant for brown eyes and G is dominant for green eyes, we want to know what's the probability, because I want my kid, I did genetic testing and I'm being facetious just to be funny, and I want my kid to have brown eyes. Okay, you need to find the right person, but okay, that's not really, hopefully it's not going to happen that way, but we want to look at and say, well, the genotypes in this individual, we want to state it a different way. What is the possibility that this offspring is going to have green versus brown eyes. And so in this individual, they have that genotype. And when we use the Punnett square, we're going to be able to figure out not only the genotypes, but the phenotype probabilities. Doesn't mean it's going to happen, but it could. So there's a 1/4 or 25% chance that they will be big A, big A. In that case, they will have brown eyes. There is a 50% chance that they can be big A, little a, big A, little a. So there's a 50% chance that they could be heterozygous. Okay? And then there is a 25% chance that they could be little a, little a. So that means that this individual has what percentage chance to have green eyes? 25. We all can do basic math. That's how Punnett square simplifies it for every genetic probability. And so the confusing part for some is, well, wait. So why did I put this out here? There is a 75% chance that they will have brown eyes. Don't just reverse it on me and go, because we already took the other 25. That's the way I do math too. But if it's a real answer we're looking for, is say, well, there's no difference between big A, or big A, little a. Big A is dominant, you're going to express it. Make sense? So in that case, that's why the phenotype and genotypes can be different. Yeah.
Okay, so 2 questions. So big A versus the 2 little A's, you would still call those homozygous, but one would be recessive and the other would be dominant. Follow-up question, why does the genotype matter if, like, to get the probability, you're still solely looking at, I guess, the phenotypes of how it expresses?
Well, we're plugging, I don't know if I caught just the question, but we're plugging the genotype into the Punnett square to determine the probabilities. That makes sense? This square is an easy way of taking this. Now, if you're very astute, like you probably are, you can look up here and probably make out that percentage without ever using that square. But this is a basic example. So. Is that what you were asking? That's just the info. This is everything you have in you, and this is the probability. Or maybe I'll say it this way. This is what you have in you, but this is what you're expressing. Because in you, it's no longer a probability. It happened already. Right? That's why I was saying, I was starting a joke and I decided to stop it. If you could go get your genetic code and actually go searching for somebody who has some of the same dominant or recessive traits you do, so that you can try to affect this square probability. Like if you want, genetic modification, if you want to ensure you have a kid with blue eyes or brown eyes.
You do it now.
Yeah, CRISPR, that kind of stuff. I'm more of a just roll the dice type. I got 5 kids, they roll the dice. I don't need genetic testing to figure that out. Right? But I'll tell you this, I'm not going to get sappy on you, but they are all beautiful. So, my kids are better than your kids. We all know that. I'm joking, because every good parent should think that way to an extent. Don't get overboard, to an extent. Okay. So we talked about normal genotypes, phenotypes, but what if you have that set phenotype, that set phenotype, but then you have something like Down syndrome? Okay, your parents didn't have Down syndrome. Just like with dwarfism, right? Dwarfism was a de novo mutation. But Down syndrome, actually they don't call it Down syndrome anymore. It's trisomy 21, but I have a nephew, brother-in-law's kid that has Down syndrome. We still call it Down syndrome. So, but in the books they call it trisomy 21. Trisomy meaning we have 3 chromosomes on the 21st chromosome. So we have 1 additional chromosome. It wasn't a de novo mutation. They've mapped this out. What actually happened is, at some point during meiosis, remember the telomeres, and we don't want things to stick together, right? I'm going to link all this back to those telomeres, because when you had separation of DNA, one of the ovum or gametes, right, a lot of times the ovum, had a chromosome that was pulled to one side, an extra chromosome that stuck to it. So you wound up with an abnormal chromosome in that end, and they got fertilized. And I'm going to link it back to telomeres, because remember, telomeres are supposedly help chromosomes from sticking together, and they also shorten with your lifespan. So that would help you to understand when you look at something like trisomy 21, why the graph for trisomy, I have it on my slides, goes significantly up once like females cross. And it includes male and females, because they only talk about the female side. Your risk of having a child with Down syndrome goes up greatly after age 35, and as you get older.
Question with that, so you're saying that males, like their telomeres, getting smaller with time, that can produce a child with trisomy 21? Or are you only looking at the female? Like when I
It
say
could be either one.
So a father who's, I don't know, in his fifth, like Robert De Niro situation, or Al Pacino, right? They're obviously older, their telomeres are significantly shorter, and they have a child with a younger woman. Could that still increase the odds of trisomy 21, like one of those conditions?
Like to have a really older male and male like
Well,
Hollywood.
you say the telomeres can, the short telomeres can
Well,
increase.
sorry, I'm bringing in. It's a great time to come in, sir. How you doing? My answer is aneuploidy, or chromosomes deleting or sticking together and adding to one side, can happen in male or female. But it's more common on the female side as they age. And here's probably the real answer. I say probably because I'm not an expert on it. Back to who had the interesting question earlier about when does meiosis continue? Remember meiosis? One, in females, starts during development of that fetus, but it's arrested for a while. So females, as they go to maturity and start to have cycles, right? They start to release eggs or ovum, right? They ovulate every month. Well, they're still going through meiosis too, and so that can happen. You're splitting genetic information still. Are males doing that? Yes, but they never really stop. That's where I'm betting a lot of that lies with female versus male. I'm not going to go read about that tonight, but normally I would, but I don't find that real interesting, but it's just a fact. But we are, anyway. Okay, so if you have monosomy, you're about to get me keep going. If you have monosomy, it means that you are missing a chromosome. If you have trisomy, you've gained one. Alright. Before we're going to get into interesting disorders at the end of this, but just because people often ask, we're not going to discuss greatly, but there is the Rh blood system. It's another blood group system. We have a lot. Right? The Rh system is one of the most complex red blood cell systems. And these are involving antigens, right? You look at all the 30 different antigens that can be involved. So we're really simplifying blood when we say your blood type A. There's a lot of variant stuff, but we're not going to go there. We're going to leave that. But we need to discuss it for one reason. So the major gene of importance, and they kind of link them together, Rh or D. You can just say, well, those are dominant. So Rh or big D is dominant, little d is recessive. And the major antigen Rh or D is a big cause of what's called hemolytic disease of the newborn. Right? Erythroblastosis fetalis. That's why we call it hemolytic disease of the newborn in East Texas, because it's easier to say. So. And here's our relationships to our genotype-phenotype. If you have the big D, you're going to be Rh positive. That's the take on there in your phenotype. If you are little d, you'll be Rh negative. We probably all understand that already from today's talk. Okay. Here's the only reason I'm bringing it up, because we're not going to talk about transfusions and things. But if you have a mother who is a first-time mother, and she is Rh negative, and she has a baby developing inside of her that is Rh positive, right? Now, where did the Rh positive come from? Dad or the sperm, however you like to say it. Okay, this baby is Rh positive, but the mother's Rh negative. It's not going to be a problem this time, okay? Because when we look at a fetus that's developing in a womb, the blood doesn't mix. I know we all think that way, that it all mixes, you got the umbilical cord, but actually certain things can't cross the umbilical cord. And one of the things is that the mother is Rh negative, the fetus is Rh positive. Because their blood doesn't mix, her body doesn't really see that baby. Meaning, I say it like that, because if your body sees a cell that it doesn't recognize, an Rh positive cell means it has an Rh negative or Rh positive antigen on that cell. Your body will go kill that, right? That's how we destroy worn out blood cells. If the glycocalyx is different, I'm going to take you out. Okay? Well, nothing's going to happen because blood doesn't really mix, like you say, when you have the baby in the uterus. But at birth, mom's blood and baby's blood a lot of times gets a little mixed, right? Some things tear, they have problems, umbilical cord. And so if mom and baby's blood are mixed at the first time she has a baby, that means that mom will be exposed to the Rh positive antigen. Is it a problem? Not yet, because the baby's born. Doesn't matter. Baby's still Rh positive, mom's Rh negative, but she was exposed to Rh positive, so what's her body going to do now? Antibodies. She's going to produce antibodies against Rh. She didn't have them before, right? She'd never been exposed to the Rh positive antigen. In a perfect world, right? So, between pregnancies, if she has another child, she's going to develop anti-Rh antibodies. There's where the problem's going to come. If she has another child, she now has those antibodies. She was born with Rh negative. She had never been exposed to the Rh positive until she had a child. Now she has antibodies. If she has another child, that's going to be a big problem because the Rh antigen, the Rh antibody that she developed is small enough, it's IgG, and that's the only time you'll ever hear me say that. I'm leaving that for micro. It is IgG, which are very, very small, and they can actually cross the placenta. And so in this case, mom is still Rh negative. If she has another baby that's Rh positive, those antibodies are going to cross the placenta and they're basically going to kill that baby. Hemolytic disease of the newborn. It's going to bleed out. Yes.
So if you had a first-time mother who develops rhesus, well, let me back this up. So first-time mother, and she has a miscarriage with no other precipitating factors. Is that likely just a flooding compatibility type thing? Not like not Rh incompatibility, but an actual blood type.
Yeah, I mean, what you're saying is, if I'm going to rephrase, you tell me if I get it right. I have an Rh negative mother who's pregnant with an Rh positive fetus, but the baby spontaneously aborts. Is that a problem with Rh factor differences? No. It'd have to be some other blood type, or it could be a genetic disorder. The body decides to terminate the pregnancy.
Just like specifically this context.
I would think there are many reasons, but the reason would not be because the baby's Rh positive. Now, are you about to say this? I would say now, they're going to give, sorry, go ahead. They're going to give this mom Rogaine, even though she hasn't had a baby yet. Because does the possibility exist that she was exposed to the Rh positive antigen and decides to get pregnant? Is that what you're going to say? There is. There is. But I was trying to explain that here before I got to the next slide. But since you brought it, good point. Everybody getting this? Next slide is just a repeat with a box to show. You don't even know everything about Rogam. Rogam, though, they're going to give pregnant moms Rogam, right, if they're Rh negative. Right? If a mom's Rh positive, do they need Rogam? No. If you have an Rh negative mom, you're going to give them Rogam. Somebody developed this, right, and if you look, it wasn't that long ago, 1968. Green means you definitely don't need to know it, it's just good knowledge. So about 10,000 babies a year, I think that's just US, I'm not sure. It may be, I don't think that's worldwide. Died from erythroblastosis fetalis, or hemolytic disease in newborn, due to this factor until they figured this out and they made Rogam. And I can simplify Rogam in that they give mom Rogam and it masks the fact that the baby's Rh positive there. Makes the baby kind of invisible to mom's immune system. If that sounds far-fetched, remember that every day of my life right now, for the rest of my life, I take immunosuppressants. So I told y'all I had a kidney transplant, and so I take immunosuppressants, and what is it doing? It does a little differently because it's not hiding it. It's suppressing my immune system so it doesn't attack that kidney. It's what it does on it. It's going to mask that the baby's there. Okay, then we get to sex. I was just thinking, I'm walking around up here, I'm just going to flat out admit it. Because I haven't digressed much today. I went and played pickleball. You ever played pickleball last? And we play every now and then. But then this kid was there this time. Some older people's friends that we know. And this kid's there. He quit his high school tennis team. He was captain of the tennis team at O'Connor a few years ago to go play pickleball. And I was like, that's what old people play at RV parks. This is before it got real popular. And now they have chicken. You ever been to Chicken and Pickle out there? Never been to Chicken and Pickle? It's all, you need to go there. It's a big party place in San Antonio. You might know more than me, I don't know. But it's like big kids, beer, drinking, and pickleball. And good food. Food's shame plug there. The point of this was, I knew when I left that I was hurting. Because this kid's good. He had like a couple of sponsored ones. But every now and then we're playing like different groups. When he's playing though, he was on my team, I thought, I gotta hold my end, because he's really good. He just kind of stands there and hits it. I don't know how he's so good, but anyway, I was running all over the place. I'm feeling it today. I'm admitting that. I'm admitting it. I lost a bunch too. But my wife's no good at pickleball. Just a fact. I hope she knows that. I'm not great either, so. But I mean, I got the serve. You can't get the serve. I mean, why are we out here? Never say anything about your wife or anybody else in the world. I always say this, that you wouldn't tell them in person if they asked you. If they asked you. I don't like to talk about other people, but if I say something and you go, well, I wouldn't say that to his wife. Yeah, I would say that to my wife. But I said it a lot yesterday. Quit dropping it and hitting it. Just hit it over the net. Over the net being key. That's another reason I'm hurting. I'm a little bit competitive. So I didn't want to lose the pickleball game. Does it really matter if you lose the pickleball game? No. Oh yeah. Yeah. Okay, here we go. A little bit of break there. Sex determination. If you look at spermatocytes, we look at sperm cells, right? They have either X or Y. Right? So when you go through meiosis, and we left it at 23 chromosomes in each one. At the end of meiosis 2, you have 4 haploid cells, 23 chromosomes each. And if we're talking about males, that means we have 4 we'll just say it now, spermatocytes. And because of the genetics, each one is either going to have an X or a Y. Because when you look at the genetic code for male, biologically male, males have an X and a Y, females have X and X. So on the male side of developing spermatocytes or sperm cells, they can have X or Y that are in there. Oocytes in females can only contain an X. And so we could go back to the Punnett square, and now you can line them up and say, well, here we have the male that has X and Y sperm cells, and female has an XX. Use your Punnett square. That boils down and tells you, well, 50% chance that your offspring will be male or female. Which we know is not always true, because I have 4 girls and 1 boy. I don't know about those probabilities. I think it has something to do with whether you're standing or on your hands or what. Y'all read that? You gotta be all sorts of, that's not really gonna change anything, but go for it. I've read that people do that. You're not gonna have a boy unless you're in the bathroom at Chicken and Pickle or something. They come up with these crazy things for how they're gonna have it. Okay. When you look, why is it important? Let's get back, serious, why is it important? Sex-linked genes. So when you look at almost all of the genes are located on the X chromosome. Right? So in general, when you say somebody has a sex-linked disorder, meaning it's being carried on the X chromosome, you're generally referring to X. Could there be a sex-linked disorder from the Y chromosome? Yes, but most of the genes are on the X chromosome. And the way you remember that, it's just a silly joke. You always go, yes, well, we finally know that our spouses are female, our spouse includes both, so females do wear the genes in the family, because they have all the genes on the X chromosome. Not many on Y. So because of that, when they study sex-linked disorders, you're generally looking at the female side of the X chromosome to see where those are coming from. Because when you have females translating, sorry, I'm fixing this. You have females that are transmitting genetic traits. Okay, everything back there. Females can transmit an X chromosome to both sons and daughters. Well, you don't have to say it that way. Females can only transmit an X chromosome because they have XX. Okay? Males can transmit an X chromosome only to their daughters. That's basic determination of what sex that individual is going to be. Because sperm cells contain an X or Y. So depending upon which sperm cell fertilizes the ovum is always going to have 2 X's, but a sperm cell can be X or Y. And so if a male transmits the X in their sperm to the ovum, that means that offspring is going to be female. That's why it says males can only transmit an X chromosome to their daughters. So that makes it possible, you probably never thought of this unless you study genetics, is X-linked inheritance then means you're going to have an absence of what they call heterogamete to heterogamete inheritance, or father to son inheritance. Hey, if dad has, let's just throw like a big example, if dad has a genetic disorder that he has in his body, whether it's being expressed or not, on the X chromosome, right? Is he going to pass that to his son? No. That's heterogamete to heterogamete, now son, non-inheritance. They won't get it. But that X is going to go to the female. If he has a daughter, they will get it. Actually, all the daughters will get it. Right. Now, let's take it one step further and then we'll move on. It's because all daughters of an affected male are obligate carriers. And the way I was going to say it is, if, I keep switching sides from male to female, as if dad passes only Y onto the male. Let's stay in the middle. Dad has a disorder for, I'm trying to make something up, 3 arms, right? And whether or not he's expressing 3 arms, let's say he has 3 arms, and it's on the X chromosome that caused that. Is he going to pass it on to his son? No. Is he going to pass it on to his daughter? Yes. Dad has 3 arms over here. With his daughter, is there a 100% chance his daughter is going to have 3 arms? No. Reason being is Dad passed 3 arms, the trait for 3 arms, onto his daughter, but she may or may not have 3 arms because who else is involved in this equation? Mom. And depending on which, out of the genotype, which phenotype gets expressed, it goes back to, we'd have to look at dominant stuff again, but we're going to leave it there. So, like I said, she's an obligate carrier then, at minimum. She's either going to carry that or express it. But she's going to pass it on. Anyway, forget about it. That's too far into genetics. What if you have abnormalities? Chromosomal abnormalities. That's where things like trisomy come in or monosomy. And so you either have a difference in number of chromosomes or structural. So they either come apart or they get stuck together. And you wind up with monosomy, trisomy. So we're going to look at several of these real quick. And also known as trisomy 21 is Down syndrome. Here's your karyotype. If you notice that boxed in red, and I want you to be able to kind of identify these so that you know what they are. There's only a couple that we're talking about. Down syndrome. But here's what the symbology, this is a karyotype, and here's the symbology. All right, well, let's write my symbology first. Symbology tells you everything you need to know. So if I wrote my karyotype or symbology there, we would say I am 46 XY. That means that I have 46 chromosomes and I have an X and a Y, representing the biological male. If it said 47 XX plus 21, that means that you have an individual that has 47 chromosomes. So we already know there's an extra one. And it's X, this person would be XX, so that's a female, 2 X's. And then the extra chromosome is going to be found on the 21st chromosome location. Could this occur at different chromosomes? Yes. Most common though is Down syndrome. One of the most common genetic disorders really. And you can X these slides that show you what they look like, but I did it just for reference. There's one of the things you'll look at, like my nephew, is they have a single palmar trees. I got to start off by saying, if you have, it's called a simian crease. I'll show you a slide in a second. If you have one, it doesn't mean you have Down syndrome. If you have it on both hands, don't come see me. Just keep it yourself. There's your chart. You're not going to have it on both hands. There's your chart for Down syndrome. It goes up. Obviously, you don't need to. I'm not going to ask you what's the probability at 40%. You can X this chart too. It's for example. There's your palmar crease, or what's called a simian crease, simian line. This is an actual student. You notice they have one straight line here, and they have separation. So they should separate like that. Having one can be normal. It is normal. You're not like partially Down syndrome. It is the variance. Okay, I'm trying to finish for the break, or because we're almost done with genetics. Klinefelter's syndrome is an interesting one. Klinefelter's is not hermaphroditism. Hermaphroditism means that you have ambiguous genitalia. I'm not going to discuss that. You have both male and female genitalia. That can occur. Don't need to go there yet. But in Klinefelter's, what they see is if you look at chromosome 23 there, the sex chromosomes, there are 2 X's and 1 Y. See? So don't read this wrong. most common human sex chromosome disorder. Those are specific words. Most common human sex chromosome disorder. Down syndrome is most common chromosome disorder. So I mean, they separated it. But even then, you don't see that. We don't see this a whole lot, right? But if you're going to have a sex chromosome disorder, this is probably the more likely one. Okay? And so this is Klinefelter's, and its symbology is 47XXY. And before even, the reason I do the slides of what they characteristically look like is so that I can go, okay, well here's Klinefelter's, and this individual would be XXY. So we have an abnormal thing going on with our chromosomes here. Right? Well, if that's the case, if there's an X and a Y present in biology, what would we consider this individual? Male. But they have an extra X. So before you even look at the next page, if somebody has an extra X, think about it this way, the way I look at it is you have an X, you have a Y, but we have an extra X. X is, if you think about puberty, female secondary sex characteristics, because they have an extra X, it's going to be someone who appears to be male, but has some more female traits because of that extra X. And that's exactly what we kind of see. And it can be even more negative than that because you can develop breast. This is biologically male but can develop breast, small testes. You don't need to know all these. Testosterone levels are low, but FSH, LH become higher. I don't care about the rest, but at the severe end, this is what I care about. This is one of the only reasons. I mean, there are many reasons, but why I would want to know if I'm treating somebody who has Klinefelter's, because as they age, because they have that second X, they're at risk for disorders you generally only see more common in females, like germ cell disorders or osteoporosis, because osteoporosis is more common in females. Sorry, I would digress more, but I have 2 slides left and I'll finish, right? Yeah. Here's Turner's syndrome. Turner's is XO. So let's decide what this person looks like. X, and there's no Y or X, right? So just looking at the symbology, 45XO means we're missing a chromosome, and it happens to be a sex chromosome that we're missing. So O is just a placeholder. That means there's no other X or Y there. That means that biologically this is considered female. Right? And if you look at this person, they're going to produce a little estrogen, they won't go through puberty. Some hallmark signs is a webbed neck, or what they call milkmaid's arms, where their arms are, if you look at her arms, they have the pubis valgus, they're going out. I guess people who did milking cows used to get that, I don't know. That's what they call it though. Heart problems, health effects. Here's the thing, I can make this back and we'll be done. I know I'm into your time, Elliot. If you have Klinefelter's, you have an extra X, you get some secondary female sex characteristics and problems that go with that. But if you're XO, you're lacking that second X. So they don't normally go through puberty, they're sterile. They don't develop a lot of the secondary sex characteristics of female men because they're missing that X. Alright, it's y'all's time. I took a little video break, so who's next anyway? Is Mike would tell her I said you can have an hour before the next class. Y'all have a good day. I'll see you tomorrow.
Would you say this is Tina Turner syndrome? Is it Tina?
Yeah. Amy Turner syndrome. I would never say that. I'm funny as Mike called me. A lot of y'all heard this, but I was